Good afternoon, class. Are you there?
Afternoon po, Doc.
Good afternoon, Doc.
Okay. Our topic this afternoon is about oxidation of fatty acids. Now, by the way, this is not included on our bi-monthly, for our second bi-monthly. This is not included. Just like also the lipids of Dr. Kapungkol is not also included. Okay, we are just utilizing this the time because we might be delayed during the second semester. Okay, all right. So let's have oxidation of fatty acids. So lipid metabolism, this is, so we have an overview. Lipids are a diverse group of compounds. It is a heterogeneous compound, unlike our carbohydrate, where it is mostly, it is carbohydrate. The carbohydrate is saccharide molecules. Now all of them, and they are soluble in water. Now this one is a diverse group of compounds, meaning heterogeneous compounds. Now that they are not related to each other, but they form as a composite of diverse compounds. Okay, and it is not soluble in water. Okay, and lipids, fatty acid, as represented by your fatty acids, provide the human body with the major source of energy. Now, we mentioned earlier in our previous studies, we said that tissues would prefer to have glucose as fuel, with the exception of your heart muscles. You know, your heart muscles, although it can use glucose as its source of fuel, they would prefer to use fatty acids. And there's a reason behind it. Because you know, your heart keeps on working, whether you are awake or sleeping. No? So there must be a constant supply of energy. And the best source of energy that can give you more ATP is your fatty acids. So that's why it says here, lipids provide human body with a major source of energy. No? So this is unique for your heart muscles. The other tissues would prefer glucose. Okay? All right. Now, you should also know that your fats provides 9 kilocalories per gram, while your carbohydrate will only give you 4 kilocalories per gram. So you have more than twice per gram if you use fatty acids as your source of energy. Okay, that's why your heart likes the fatty acids. So it's 9 kilocalories per gram. Okay. obtained from the diet or are synthesized in the liver after a meal. They are transported in the blood by lipoproteins. This is the carrier, because we know that they are insoluble in water, and you know blood is an aqueous solution, so they have to have a carrier. So we have here a protein with a lipid, so it becomes a lipoprotein. No? And then this is stored in the adipose tissue. Adipose tissue. Okay. The major classes of blood lipoproteins include chylomicrons, VLDL, IDL, LDL, and HDL. We will be talking about this later on. In addition to your triglycerides, now by the way, triglycerides and triacyl glycerol, just the same. Now, the old books uses triglycerides. The new books uses triacyl glycerol, because triacyl glycerol is more descriptive than when you say triglycerides. No? Tri is 3. Acyl, fatty acid. No? Acyl is acid. So 3 acids attached to a glycerol. So that is triacyl glycerol. So that's why it is more descriptive rather than triglycerides. But they are the same. No? They are the same. Okay. So the triacyl glycerols, now in addition to the triacyl glycerols, they contain proteins, phospholipids, cholesterol, and cholesterol esters. These lipoproteins that we have here. Okay. The triglycerides or the triacyl glycerol of chylomicrons are from the intestine. No, intestinal cells, from the dietary lipids, the one that you eat. And the triacyl glycerols of VLDL, so you see triglyceride or triacyl glycerol of chylomicrons, meaning coming from the intestine. The triacyl glycerol of VLDL is from the liver. Okay? Produced in the liver from dietary constituents, mainly, look at this, mainly carbohydrates. So, you have here a hint already that if you have extra rice, no, the extra rice will be converted into triacyl glycerol. Okay. The IDL is an intermediate, okay, intermediate density lipoproteins, and LDL, low density lipoproteins, are produced in the blood capillaries by the digestion of triacyl glycerols of VLDL. HDL is from the liver, transfer and activator of lipoprotein lipase to chylomicrons and VLDL. They also carry cholesterol from the peripheral tissues, no, meaning the tissues far away, no, peripheral, and bring them to the liver. That's why this is known as your good carrier for cholesterol. And they just shorten it, they just say good cholesterol. While this one, LDL, also carries cholesterol, but it is known as the bad cholesterol. Why is this bad and why is this good? Huh? It is bad because LDL spreads out the cholesterol into the peripheral tissues. So nagkalat ng cholesterol. While this one, HDL, removes the cholesterol from the peripheral tissues and bring it to the liver. So he gather all those cholesterol from the outside and bring it to the liver to be oxidized, no? To be used in the formation of bile acids. And bile acids are excreted through the intestine and goes out with your feces. So that's how it is, no? That's how you exit your cholesterol from the body. Okay. The triglycerides or triacyl glycerol of chylomicrons and VLDL are hydrolyzed by a lipase known as lipoprotein lipase, meaning a lipase acting on lipoproteins. What are those lipoproteins again? Chylomicrons, VLDL, HDL, LDL. Okay, so the lipoprotein lipase acts on those lipoproteins to release fatty acids and glycerol. The fatty acids may be taken up by various tissues and oxidized. This is our topic for this afternoon. Okay. In the adipose tissue cells, these fatty acids are converted to triacyl glycerol again and stored. During fasting, however, fatty acids are derived from the breakdown of triglycerides that are being stored in the adipose tissue and can be oxidized through a process known as beta oxidation of fatty acids to form ATP, carbon dioxide, and water. Okay? So various tissues producing energy in the form of ATP, carbon dioxide, water, and ATP. In the liver during fasting, fatty acids are converted to ketone bodies. No? It is not only by fasting that fatty acids are converted to ketone bodies. In certain conditions, like uncontrolled diabetes mellitus, the fatty acids are converted to ketone bodies. So two conditions where your fatty acids are converted to ketone bodies are fasting. So during fasting, the stored fatty acid in the form of triacyl glycerol or triglyceride are converted to glycerol and fatty acids. And the fatty acids are oxidized. No? And of course, if there's so much of it, the oxidation, you will form ketone bodies. Okay, that's during fasting. In diabetes mellitus, there's plenty of glucose, but the glucose cannot get inside the cell. So the cells are starving. So it tells the body that we are starving. No, it is just like you are in the middle of the ocean. There's plenty of water around you, but you cannot drink it. No? So that's what happens. And so the body results or go into the breakdown of fats, thinking that there is no glucose because the cells are clamoring for a fuel for ATP. But since glucose cannot get inside the cell, so they send signals. And the signal are received and fatty acids are released from the breakdown of triacylglycerol in the storage or in the bodega. Your bodega is your adipose tissue. Excuse me. Excuse me. All right. So, your ketone bodies are utilized and are taken up by the muscle, the kidneys are oxidized. So you still have energy. By the way, gluconeogenesis. When do we have gluconeogenesis? When can we have gluconeogenesis? Okay. Gluconeogenesis cannot proceed if there is no fuel. And what is the fuel that is to be converted into ATP is fatty acids. Always remember that. So gluconeogenesis is dependent now of beta oxidation of fatty acids. Okay. Lipids also, in our study of membranes, is a component of your cellular membrane. Membranes contain phosphoglycerides or acylglycerol and sphingolipids, which are both hydrophobic, which contains hydrophobic and hydrophilic regions. Cholesterol is also present in the plasma membranes. Lipids, therefore, have many diverse functions. Bile salts are made from cholesterol, steroids as well, which are derived from cholesterol, fat-soluble vitamins, and compounds such as prostaglandins. Okay? All right. Now, how is triacylglycerol digested? So in man, the dietary fat is triacylglycerol, obtained from the bodega of your adipose tissue, in animals and also in plants. Okay. The dietary triacylglycerol, which are water insoluble, are digested in the small intestine to fatty acids and to monoacylglycerols. These digestive products are resynthesized in the triacylglycerol intestinal epithelial cells and secreted chylomicrons. Okay. Before we go into that. So the enzymes that is responsible for digestion of your triacylglycerol, dietary triacylglycerol, is lipase. Okay. You have several lipases. You have lingual lipase. Then you have gastric lipase. Then you have pancreatic lipase. And then you have intestinal lipase. You see? All right. Our lingual lipase is rudimentary. So it is not working now. Probably in our evolution, when we were not so evolved, we have that. Now, Some mammals has this lingual lipase. Like for example, rodents. The rodents, mice and mouse. Mice, no, mouse. Rats, no? Okay. Now, if you notice how the rats or the mice eat, no? They get a bite of the food and then they are chewing it for a long time before swallowing it. You see, that is by nature. No? That's what I've been telling you, that you should chew your food properly. Now also, doing this, the lingual lipase of the rats and mice, no, is secreted out. And so there is already a digestion there in the mouth. The longer it stays in the mouth, the more digestion is going on. Aside from the salivary amylase, which acts on carbohydrate. Now, our salivary amylase is still intact, but we do not have the lingual lipase anymore. Now, through evolution, this was removed. Perhaps our intestinal lipase or our pancreatic lipase are very, very efficient. That's why there is no need for it, for us humans. All right. Now, what about gastric lipase? Okay. When you were born, when you are an infant, your pancreas, the pancreatic secretions are not so well in place yet. So, the fats that are found in the milk that you are subsisting as an infant has to be digested well. And because the pancreatic lipase is not so well in place yet, there is what we call gastric lipase. Now, this gastric lipase, it is in the stomach. So we only find this in infants, because once you become an adult, this gastric lipase secretion stops. No, it stops. This gastric lipase. All right. So once the food goes into the small intestine, we know that this is the pancreatic juice. No, and this one of the enzymes there for fats is your pancreatic lipase. Okay, and this is also aided by an intestinal lipase found in the intestine also to help in the breakdown of the triacylglycerol into fatty acids and glycerol. Did you get it? All right. And then once in the chylomicrons later on, as it passes through into the blood, and there is what we call a lipoprotein lipase found in the capillaries. Okay, so there's another lipase. That is your lipoprotein lipase, which acts on the lipoproteins. VLDL, no, chylomicrons, those are your lipoproteins. All right. And fatty acids are released and oxidized in various tissues or stored as triacylglycerol in the adipose tissue cells when your cells does not need any more the fatty acids because they are fully charged. There's plenty of ATP. Okay. All right. So we have it here. Major lipids in the diets are triacyl glycerols and to a lesser extent phospholipids. They are all hydrophobic compounds and have to be hydrolyzed and emulsified to very small droplets in form of micelles, which is about 4 to 6 nanometer in diameter before they can be absorbed. The fat-soluble vitamins like A, D, and K, and a variety of other lipids, including cholesterol and carotenes, are absorbed, dissolved in the lipid micelles. Absorption of carotenes and fat-soluble vitamins is impaired on a very low-fat diet. Okay, those who are taking multivitamins and you do not want to eat fatty, oily, greasy food, no, you will have a hard time absorbing the fat-soluble vitamins A, D, E, and K. Okay? All right. Hydrolysis of triacyl glycerols is initiated by lingual and gastric lipase. Okay. We said that this lingual lipase is no longer functional when you are an adult, as well as your gastric lipase. There might be a little remnant of your lingual, through our evolution, but it is negligible. It's negligible. But the gastric lipase in infants is very, very helpful. Very, very helpful. Because the pancreatic lipase is not yet well in place when you are an infant. That's what we have been saying, no? Okay. All right. We will leave this. We will go to the diagram. Okay. So you will understand better. Is the diagram here can you see this diagram? Okay. All right. What is this? Can you recognize that? Okay. That carbon 1, carbon 2, carbon 3 of glycerol. So this is in the form of capital letter E that symbolizes glycerol. No? Okay. Carbon 1, carbon 2, carbon 3. Now, triacyl glycerol. So you have acyl, 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 or fatty acids. No? So a fatty acid is found at carbon 1, a fatty acid at carbon 2, a fatty acid at carbon 3. So you call it triacyl, 3 fatty acids attached to glycerol. The attachment here is an ester linkage, and the process is known as esterification. Okay? All right. So now this is now in the intestine. This bar here and this one represents the wall of your intestine. This is the lumen of the intestine. So this is the wall here, another wall here, and this is cut lengthwise, and so this is the lumen of the intestine. All right. So you have your pancreatic lipase because this is now in the intestine, in the small intestine. Okay, pancreatic lipase. It removes fatty acid at carbon 1 or carbon 3. So the external fatty acid, it cannot remove the fatty acid at number 2 position. So it's only the external That is carbon 3 and carbon 1. So as you can see it here, the pancreatic lipase removes this fatty acid at carbon 3, releases as free fatty acids, and what do you have? You have 1,2-diacyl glycerol because there is no more fatty acid here. No? Can you see that? All right. So this 1,2-diacyl glycerol is acted upon by the same enzyme, pancreatic lipase, this time removing the fatty acid at carbon 1. All right. So what is left is a fatty acid there at number 2. Hydroxyl, hydroxyl there. So here's the fatty acid, the other fatty acid. And this fatty acid now goes inside, penetrates the wall into the wall of this I'm sorry, I said lumen. It's not lumen. This is the cellular tissue. Huh? Huh? Okay. So inside the intestinal cell, this fatty acid is activated because that's free fatty acid. It is activated by an enzyme, acyl-CoA synthase. And what is the result? Is acyl-CoA. Acyl-CoA. All right. If the fatty acid is stearic acid, once it is activated, it becomes known as stearoyl-CoA. If it is palmitic acid, it becomes palmitoyl-CoA. Activated, huh? Okay. Oleic acid becomes oleal-CoA. Get it? All right. Now, so that is now this here, 2 monoacyl glycerol, 1 fatty acid is left, is now absorbed. Look at that. And of the total 100% triacyl glycerol, about 72% are absorbed in this manner. There you are. So the 2 monoacyl glycerol is there. Inside now the cellular, the cell of the intestine. Okay? All right. So what happens next? This activated fatty acid, acyl-CoA, now esterifies this hydroxyl here and this hydroxyl here to reform back to triacyl glycerol. You see that? That's the same thing here. That's the same thing. There you are. So, well, why do we have to do this? Now, removing the 2 fatty acids from your triacyl glycerol makes the molecule smaller. Okay? And the wall of the small intestine now can allow that to come in. Okay? And this is reconstructed back. The fatty acid has been activated, no, and put back. You see that? No? So we will stop it there. We will not continue going this way. But let's look at this here. Now, if now this fatty acid is now transferred. Okay, look at this. The fatty acid at the number 2 position is transferred to number 1. That's the one there. Through this enzyme isomerase. And what do you have? 1 monoacyl glycerol. Because this is similar to that, only that the fatty acid is found at number 1. It is still absorbable. And this is absorbed, no, about uh, a very small percentage, okay? Very small percentage, about 6%, okay? And once it is inside, the 1-monoacyl glycerol, okay? And the fatty acid that is already activated now will esterify these two hydroxyl groups, and they become also triacyl glycerol again, just like that one. You see? All right. So that's only about a very, very small percentage. However, this 1-monoacyl glycerol can still be, because the fatty acid is there already at the periphery, the pancreatic lipase can remove that. And you have now triacyl glycerol. Oh no, I mean glycerol only. And the glycerol is absorbed 22%. There you are. And of course, the fatty acid goes this way and that way. Okay? All right. So you can see that the glycolysis of the cell can also look at that. Esterify, no, glycerol 3-phosphate, and then this esterify this fatty acids, this hydroxyl here with fatty acids, and then it becomes triacyl glycerol. Okay? So the idea here of the digestion in the stomach, or in the intestine, small intestine, is just to make this smaller so that it can get inside intestinal cells. Now, once they are reformed back to triacyl glycerol, they can now come out on the other side, because you have here lymphatic vessels. Lymphatic vessels. Okay? Now, since this is insoluble in water, and lymph is aqueous solution, they cannot go directly to the lymph. They have to be packaged. They have to be packaged. And the packaging here now is known as a lipoprotein. What kind of lipoprotein? Coming from the intestine, it is known as chylomicron. Chylomicron. There you are. Chylomicron is the packaging of your triacyl glycerol so that it can go inside the lymphatics together with the lymph. And these lymphatic vessels in the small intestine, they coalesce, because there are many of them, they coalesce together to become a bigger duct. And we call this duct, no? The thoracic duct. You see? It goes, with this duct penetrates the diaphragm, goes up through the thorax, right into the left side of the body, and it connects with the left subclavian vein. So the lymph now mixes with the blood. Okay? That is how your triacyl glycerol absorbed from the small intestine goes into the blood. It is the chylomicron packaging, goes into the lymph, the lymphatic vessels. The lymphatic vessels coalesce to become bigger duct, known as the thoracic duct, and the thoracic duct goes up to the left side, no, and in at the base of your neck, you have your left subclavian vein. And this thoracic duct now empties into the left subclavian vein. And so now your chylomicrons is already in the blood. You get it? Huh? And this is also, it is not only the chylomicrons carry this. The chylomicrons can carry also absorb vitamins A, D, E, and K, and goes with that. As well as the cholesterol that are absorbed here and goes also with the chylomicrons and goes directly now into the blood. The chylomicrons are there. No? It carries. The chylomicrons carry cholesterol, fat-soluble vitamins, okay, and triacyl glycerol. Okay. So you know now how my battery is weak. Excuse me. There you are. Okay. So that is how your triacyl glycerol now goes into the blood. And the blood, of course, in the small capillaries, you have enzymes. Lipoprotein lipase interacts with the chylomicron, digests the triacyl glycerol, and releases fatty acids as well as glycerol into the blood. The fatty acids are taken up by peripheral cells. And so these peripheral cells now utilizes the fatty acids as a source of energy. For example, your heart muscles, they like it so much. No? So that is now how your fatty acids are in the blood. Okay? So you have there within the intestinal cells, the monoacyl glycerol hydrolyzed to fatty acids and glycerol, and two monoacyls are recycled to triacyl glycerol via the monoacyl glycerol pathway. So recycled, no? Recyclated, okay? Glycerol released in the distal lumen is absorbed into the hepatic portal vein, and the glycerol released with the epithelium is reutilized triacyl glycerol synthesis via normal phosphatidic acid pathway. When we will be able to see this in the synthesis of fatty acids. Long-chain fatty acids are esterified to triacyl glycerol in the mucosal cells, together with other products of lipid digestion secreted as chylomicrons into the lymphatics, entering the bloodstream via the thoracic duct. Short and medium chain fatty acids are mainly absorbed into the hepatic portal vein as free fatty acids. Okay, how far are you in your anatomy? You are not into circulatory yet, no? You have not seen this thoracic duct yet? Anyway, this is quite a big duct, and it can be seen. It penetrates the diaphragm as it goes up through the thorax. and into the left subclavian vein. Okay. Cholesterol absorbed, dissolved in lipid micelles, and so on and so forth. Okay. All right. Importance. What we had was just only a, what you call this, overview, no? So fatty acids are broken down in mitochondria, acetyl-CoA, and generates amount of energy. They form, when there's so much of this, there's so much of oxidation of fatty acids, you will form these ketone bodies, acetoacetate, hydroxybutyrate, and acetone. Acetoacetate and hydroxybutyrate used as fuels by extrahepatic tissue in normal metabolism. But overproduction of ketone bodies causes ketosis. Now, since ketones, when they become high in concentration, it is acidic. So you do not have only ketosis, but also ketoacidosis. Ketoacidosis. So it says here, ketone bodies are acidic. So if there's plenty of it, you will have now ketosis and acidosis. So you call it ketoacidosis, which is, of course, fatal. Because gluconeogenesis is dependent on fatty acid oxidation. So I've mentioned this already, that your gluconeogenesis is dependent on oxidation of fatty acids. Because they need the energy to form glucose out of non-glucose materials. This occurs in various stages of carnitine deficiency. Now I'll be talking about this. Carnitine deficiency is now I don't know if you know carnitine. Anybody knows what is carnitine? Have you not drink pineapple juice? Huh?
Yes, Doc.
Oh, what did it say in the label? Did not say any, it did not say anything about the pineapple?
Rich in vitamin C.
Well, aside from that, there is a very important
L-carnitine.
Carnitine. Yes, carnitine. And what did this is advertised, the pineapple juice, that if you want to reduce, you take pineapple juice because of the presence of carnitine. So what's the importance of that carnitine? Now this is
Bawal.
where it comes in. The carnitine here, because this is the compound that will allow your fatty acid to go into the cell. Okay? It is, an analogy to that is your insulin, no, which allows glucose to enter the cells. Okay? So that's carnitine. And this is found in pineapple juice. That's why they advertise it that way. To, if you want to reduce, drink pineapple juice. No? Okay. So, if there is a deficiency of that, no? So, inhibition of fatty acid oxidation. inhibition. Okay, and this is the enzyme, carnitine palmitoyl transferase. We'll be looking at these enzymes in the diagram later on. Okay. Oxidation of fatty acids occurs in mitochondria. Although acetyl-CoA, now, wait a minute. What other processes have we seen inside the mitochondria?
Cycle.
Krebs cycle, very good. And what is the substrate of the Krebs cycle? Allah. Unsay substrate sa Krebs cycle? Acetyl-CoA. Very good. Acetyl-CoA. So there you are. Acetyl-CoA. Huh? Acetyl-CoA. Now, the fatty acid metabolism is metabolizing the fatty acid by removing 2 carbons at a time, converting it into acetyl-CoA. So for example, you have stearic acid, 18 carbon atoms. How many acetyl-CoAs can we make out of it? Eighteen carbon atoms divided by 2, pila?
9, Doc, 9.
9 acetyl-CoA. Sus, kadugay. I said acetyl-CoA, 2 carbons. When I said 2 carbons at a time. So you remove 2 carbons at a time. Now you have 18 carbons. So how many acetyl-CoAs? That's 9. If it is 16, like palmitic acid, how many? 8. Diba? Uy. Ay, na nga, mahina kayo sa math. Sabi nila mga waray magaling sa math. Diba? No? Now. Okay. Now, acetyl-CoA is really very versatile. It says here, acetyl-CoA is both the endpoint of fatty acid oxidation, meaning the breakdown of fatty acids. And at the same time, it can also be used as a starting substrate for fatty acid synthesis. Huh? No? Can you see this? All right. Question. In fasting, what did you say? What did we find in fasting? There is a breakdown of triacylglycerol. The fatty acids are being oxidized. No? Now, during fasting, can we use, it's easier, can we use fatty acid as a starting substrate for the synthesis of fatty acids during fasting? Hala, pila mo kabuk? 100? How many are you? Oray good isa? Come on, think. During fasting, because it's easier, that acetyl-CoA can be is used as a starting substrate for the synthesis of fatty acid. But it is also the end product of the breakdown or catabolism of fatty acids. Now the question is, during fasting, there is an increased oxidation of fatty acid, forming a large amount of acetyl-CoA. And we just have said that if there's a large amount of acetyl-CoA, there is a probability of ketosis, correct? It says there, di ba? Now, the question is, can we synthesize that acetyl-CoA? Because there's plenty of acetyl-CoA. Can we synthesize it into fatty acids during fasting? Why? Yes, fatty acid, acetyl-CoA is formed in the mitochondrion, but it can always go out fatty as disguised as other compound and goes into the cytoplasm. That's correct. No? The answer is no, you cannot. Because what is in fasting? Why does the fatty acid being broken down in fasting? To produce ATP, yes. But why? Why it is using fatty acids? Yes, for gluconeogenesis. Now the answer there is, when there is now a need for glucose and it is absent because you are fasting, breakdown of fats occur.
Ketogenesis.
And ketogenesis follows. But why can't we synthesize it back into fatty acids? The extra acetyl-CoA, so that you will not have ketosis. What is absent? Hala.
Because the amino acids can't be used to cannot be synthesized back to glucose, Doc?
Okay, I'll give you a hint. This has got to do with hexose monophosphate shunt. NADPH. What is the use of NADPH? Hindi, for synthetic purposes. When you synthesize, I've been emphasizing that. Hexose monophosphate shunt has only two products. What are those? NADPH and ribose 5-phosphate. Yes. What is the function of NADPH? Biosynthesis of what?
Nucleic acid.
I told you before. Fatty acids, I've said, you cannot have fats by eating fats. I meaning to say you cannot have fats stored by eating fats only. There is the need of glucose. Because glucose has NADPH, and this is the one that put together the acetyl-CoA. NADPH. And even cholesterol, you cannot have cholesterol without NADPH, without glucose. And when does hexose monophosphate shunt becomes operational? Oh, high glucose. There you are. So high glucose, extra rice. What do you have? Fats. As a result. Why? You can put together now the acetyl-CoA together to make it into a chain of fatty acid. Or you can form cholesterol out of the acetyl-CoA because cholesterol is made up of acetyl-CoA. Put together again in the presence of NADPH. Without NADPH, you cannot form it. So now, you do not blame the fat for becoming fat. And you have to blame your carbohydrate. Diba? Okay. So that's why your acetyl-CoA here is the end product of fatty acid degradation or catabolism. And it is also the starting substrate for fatty acid synthesis. That's correct. But in two different situations. The breakdown of fatty acids, meaning you are fasting. And if you are fasting, there's no glucose. Where you do not have, you cannot have the formation of NADPH from hexose monophosphate shunt, because hexose monophosphate shunt will only work or becomes operational when there is plenty of glucose. We said hexose monophosphate shunt is a manner of oxidizing glucose without producing ATP. And it produces only two compounds or products, and that is NADPH and ribose 5-phosphate. Ribose 5-phosphate for the making of nucleotides to make it to nucleic acid. But what is important is your NADPH for fatty acid synthesis and cholesterol synthesis. See? It's your carbohydrate that makes you fat. It's the carbohydrate that makes so much cholesterol. And we are barking at the wrong tree. No? There was a time in the 1980s that there was a legislation in the US to minimize the intake of fats in order to lower down the incidence of atherosclerosis and other cardiovascular disorders. And so everybody was trying to avoid fat. But yet, atherosclerosis was still on the rise, cardiovascular diseases are on the rise. So it is not, therefore, the fat. It is something else. And they found out that it is the sugar, glucose. Now there is another, is your high fructose syrup. And you know fructose can easily become glucose through isomerization. And utilization of fructose usually goes into glycolysis, but it bypasses some steps in glycolysis so that fructose can easily be used faster. use fructose faster. And so the problem is still there now because of this high fructose syrup. High fructose syrup are now found in almost anything processed food. Even your ketchup, high fructose syrup. All your soda drinks, high fructose syrup. Or anything that is sweet, it's now using high fructose syrup. Why? Because it is very cheap compared to sucrose, which is taken from sugar cane. It's expensive. Or sucrose from sugar beets. Huh? So there you are. Now they have found out that it is not really fats that make you fat. It's the carbohydrates. It's the sugar. So minimize your sugar, you minimize also your salt. Huh? All right. Let's go forward. Now, what I'm trying to tell you now is I'm connecting these things from your carbohydrate now into your lipids. There is a connection. Okay? All right, so it's there. Breakdown of fat is not simply the reversal of the biosynthetic pathway because both are, the two are found in separate areas. Fatty acid synthesis is in the cytoplasm. The fatty acid breakdown or catabolism is inside the mitochondria. Okay? So the separation of fatty acid synthesis, oxidation, mitochondria from biosynthesis in the cytoplasm allows its process to be individually controlled and integrated with tissue requirements. Each step in fatty acid oxidation involves an activated fatty acid known as acyl-CoA. It's catalyzed by separate enzymes utilizing NAD and FAD. It is not NADP, NAD and FAD as coenzymes. Generates ATP. It is aerobic, requiring the presence of oxygen. That means it is utilizing the electron transport chain or the respiratory chain, inner membrane of your mitochondria. All right, here, fatty acids are transported in the blood as free fatty acids. Free fatty acids. When we say free fatty acids, they are unesterified. No? So it is FFA, sometimes it is called UFA or NEFA, non-esterified fatty acids. Fatty acids that are not esterified. In the plasma, the long chain free fatty acid is combined with albumin. Why is this? It's because long chain free fatty acids are not soluble in water. And you know that your plasma is an aqueous solution. So it is in combination with something that is soluble in water, and that is albumin. So your albumin carries the longer chain fatty acid like palmitic acid, stearic acid, oleic acid, so all of those. Okay. And in the cell, once you get inside the cell, since the cytoplasm is also an aqueous solution, you have another binding protein there, and it is called fatty acid binding protein. So, in fact, they are not really free because they are always in combination with a protein. However, the shorter chain fatty acids are more water soluble and exist as the unionized acid or fatty acid anion. Like short chain, 6 carbon atom, 4 carbon atom fatty acids, or 8 carbon atom fatty acids. They are short chain, and then you have your medium chain. And your medium chain is up to about 12 carbon atoms. Okay? Fatty acids are activated before being catabolized. And it is catalyzed by this enzyme, acyl-CoA synthetase. Meaning to say, the fatty acid reacts with coenzyme A, and this is the enzyme that catalyzes it. And you have now an activated fatty acid known as acyl-CoA, or active fatty acid, using one high energy phosphate and forming AMP. No? The PPI is hydrolyzed by an inorganic pyrophosphatase, and the energy is given to the reaction. So there is a loss of further high energy phosphate, ensuring that the reaction goes to completion. So acyl-CoA synthetases are found in the endoplasmic reticulum, peroxisomes, and on the outer membrane of the mitochondria. Oh, here we are. Now, you have this outer membrane of the mitochondrion, inner membrane of the mitochondrion. So you have that, the two membranes, outer and inner. So out there is the cytoplasm, and here is the matrix. This is your intermembranous space. All right. So you have here free fatty acids, FFA, coenzyme A plus ATP. Through your enzyme acyl-CoA synthetase, you activate the fatty acid and becomes acyl-CoA. The ATP becomes AMP and a pyrophosphate. Acted upon by pyrophosphatase, that will be 2 inorganic phosphates. And the energy release still utilized in this. So in this reaction, you are actually utilizing 2 high energy phosphate bonds. 2 high energy phosphate bonds. Okay? Now, this outer membrane of the mitochondrion is not too selective, because other membranes will not allow a compound containing a coenzyme A. So the acyl-CoA can penetrate this because it is not really very restrictive. Okay. However, this membrane, the inner membrane, will not allow acyl-CoA to pass through. It will not allow. So, what are we going to do with this? We have to remove the CoA. So, removing the CoA, you have what we call carnitine. So, here we are. Here is our carnitine from pineapple. Carnitine palmitoyl transferase. All right. So you have your carnitine and acyl-CoA reacting, removing the CoA, and then you have your acyl-carnitine. Now, with the carnitine, that fatty acid now can come in through the help of this enzyme, a translocase. Now, carnitine, acyl-carnitine translocase. So here is your acyl-carnitine now inside the matrix. Okay. And then it reacts because we need acyl-CoA. It reacts with coenzyme A through an enzyme known as carnitine palmitoyl transferase 2. This is different from this one. That's why this is 1, this is 2. The figure here is like that. And the figure here is different. Okay? That is 1 and this is 2. The 2 is located inside the inner membrane. And so, carnitine is removed and coenzyme A is added and you have acyl-CoA. Activated fatty acid. No? So, if only this one will allow acyl-CoA to come in, there is no need for carnitine. So there you are. Now acyl-CoA is now ready to be catabolized, chop into several acetyl-CoAs. No? Acyl-CoA is different from acetyl-CoA. Acetyl-CoA is 2 carbons. Acyl-CoA, we do not know how many carbons. No? It all depends on what is the starting fatty acid. So this goes now into beta oxidation where it is being oxidized by removing 2 carbons at a time, converting the 2 carbons into acetyl-CoA. So the role of carnitine in the transport of long-chain fatty acids through the inner mitochondrial membrane. You get it? Okay, so let's proceed. Oh, there you are. Oh. Long-chain fatty acids cross the inner mitochondrial membrane as carnitine derivatives. Carnitine is beta-hydroxy trimethyl ammonium butyrate. Beta-hydroxy gamma trimethyl ammonium butyrate. This is the one. Is widely distributed and is particularly abundant in muscles. And long-chain acyl-CoA or free fatty acids cannot penetrate the inner membrane of the mitochondrion. In the presence of carnitine, however, carnitine palmitoyl transferase 1 located in the outer membrane transfers long-chain acyl group from CoA to carnitine, forming acyl-carnitine and releasing coenzyme A. acyl-carnitine now is able to penetrate the inner membrane to gain access to the enzymes of the beta oxidation system via the exchange transporter known as a translocase, carnitine acyl carnitine translocase. The transporter binds acyl carnitine and transport it across the membrane in exchange for carnitine. The acyl group is then transferred to coenzyme A so that acyl-CoA is reformed and carnitine is liberated. This reaction is catalyzed by carnitine palmitoyl transferase 2, which is located on the inside of the inner membrane. Okay, so this is how it is. This is your this is your fatty acid. So palmitoyl-CoA or palmitic acid with the coenzyme A. This is 16 carbon atoms. Let us count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. And beta oxidation is oxidizing the beta carbon. And what is the beta carbon? The beta carbon is the carbon, no? It's the carbon right next to alpha. And the alpha carbon is the carbon next to the carboxyl carbon. This is the carboxyl carbon. That is the methyl carbon. That's the carboxyl carbon there. And right next to it, through the Greek letter alphabet, that is alpha and that is beta. This is supposed to be in the Geneva numbering as carbon 2. And this one is carbon 3. Because Genevan numbering starts with the carboxyl carbon. And that's carbon 1, carbon 2, carbon 3, carbon 4. No? So, beta oxidation meaning you are oxidizing this carbon, this number 3 carbon. Cutting this off and oxidizing this back into a carboxyl group with a coenzyme A. Okay? No, you have seen the squiggle there, high energy band there. Okay, so there will be successive removal of acetyl-CoA. So it is being chopped, 2 carbons at a time. There. So carbon, that's carbon 2. No? That's carbon 1, carbon 2, that is released. So this one becomes carbon 1, that's carbon 2. This becomes carbon 3. That's the carbon 3 there here now, the beta carbon. Then that is cut off and become acetyl-CoA. Okay? Until you will have 8 acetyl-CoAs, 16 carbon atom palmitic acid. The beta oxygen cycle generates FADH2 and NADH plus H. The several enzymes, collectively known as fatty acid oxidase, are found in the mitochondrial matrix or inner membrane adjacent to the respiratory chain. This catalyzes the oxidation of acyl-CoA to acetyl-CoA via the beta oxidation pathway. The system proceeds in cyclic fashion, which results in degradation of long fatty acid to acetyl-CoAs. Large quantities of the reducing equivalents FADH2 and NADH are generated, used to form ATP by oxidative phosphorylation. Okay. Oh, here we are now. This is our beta oxidation. Is this clear? You can refer this in your book. Oh. Okay. This is your membrane, outer and inner membrane. Okay. This course is of course outside, no? Fatty acid converted to fatty acid CoA or acyl CoA through your acyl CoA synthesis. Okay, so you have through the process, you have the two outside and inner membrane, which allows your acyl CoA to come in through your carnitine transporter, and you have your acyl CoA. So let's start here. Acyl CoA is being oxidized, and the acceptor of hydrogen is FAD. And you have here FADH2, which goes into your electron transport chain, producing how many ATPs? How many ATPs? FADH2. Allah. 1.5 now. 1.5. Oh, there you are, 1.5. Then you have your NOL-CoA. It is hydrated, hydratase, so that you will have an OH. No? And you have your 3-hydroxy-acyl-CoA. Remember that 3 is your beta carbon. It is oxidized again by a dehydrogenase. And this time it is NAD as the acceptor. And you have NADH. That NADH goes to the respiratory chain right away. You know, you should remember that this is inside the mitochondrion. And in the inner mitochondrial membrane is your respiratory chain. So this can easily go there. The FADH2 can easily go there. No? As well as your NADH plus H. They can easily go there. Electron transport chain. One molecule of NADH plus H produces how many ATPs? $2.5. Just kadugay. 2.5. So how many now? 1.5 plus 2.5? 4.
4.
4. Okay, I have 4.
For
In one
the
cycle. Oh, in one cycle. There. Huh? And then, you form acetyl-CoA man. This acetyl-CoA. Where does this go?
Krebs cycle.
Oh, Krebs cycle. How many ATPs will be formed? Canadian is a Krebs pila. Mo say 1. Taon sa inoon sabi ako.
10.
10. There you are 10. Gikan dire. 1.5, 2.5, plus 10, 14. Amo na? Usa lang ka acetyl-CoA diha. Nagikiputol. And then, okay, this is this fatty acid is minus 2 carbons, in the form of acetyl-CoA. So supposing this is palmitic, which is 16 carbons, the result here will just only be how many carbons? Minus 2? 14 na lang. And then it goes back here with 14 carbons, and it goes the same way, and it becomes 12 carbons, then go back, the 12 becomes 10 when it reaches here, and then the 10 goes back, and then when it reaches here becomes 8, and so on. So it is being chopped 2 carbons at a time. Can you get it? Huh? So that's why fatty acids produces more ATP. Oh. So you have here long chain acyl-CoA is cycled through reactions number 2 to number 5. So number 2 there, because that's number 1 there on top. That's number 2 reaction, number 3, number 4, number 5. So from number 2 reaction to number 5, acetyl-CoA being split off each cycle by an enzyme here, thiolase. There you are, thiolase. Reaction 5. Okay. So this is just the narrative for this one here. That's the narrative there. Now, if, because this is even numbered, supposing you have an odd number fatty acid because you are eating ruminants, cows, carabaos, no, goats. Those are ruminants, and they can produce odd numbered fatty acids. So you will have 13, 15, 17, no? Okay. So the last cycle will contain only 3 carbons, and that will become a propionyl CoA. Propionyl CoA from an odd chain fatty acid. And it is the only part of a fatty acid that can become glucogenic. So there's nothing in the fatty acid that you can convert to glucose except when the fatty acid is odd chain fatty acid, because the last 3 carbons becomes propionyl CoA. Where does propionyl CoA comes in gluconeogenesis? Laka. One of the questions in the bi-monthly. Huh? Well, good. Well, gi makatagaan. Balik, tanawa ko no inyong propionyl CoA. Tanawa ko no. Sa gluconeogenesis. Where will it come in so that it can become glucose? Ay, o kariya mani o. The 3 carbon propionyl CoA residue remains, and it is converted to succinyl CoA. Asa man dapitang succinyl CoA sa gluconeogenesis. Ay, nakalimtan na. Krebs cycle. Oh, musud sa Krebs cycle. Kaya gluconeogenesis incorporates the Krebs cycle. The intermediates there can become glucose. And succinyl-CoA is one of the intermediate. So it can become glucose. Did you get it? Allah? No? So this is a good question for the bi-monthly. There's tubangan nila, this compound is converted to succinyl-CoA, a constituent of the citric acid cycle. See figure 16 to mo katog diha kay so oxidation of fatty acid produces a large quantity of ATP. That is why your heart muscles like so much fatty acids as a source of energy, because it can produce a large amount of fatty acid per molecule. In comparison to glucose, there are only few. No? Oh, computation nila. Oh, that's it. Oh, so generation of ATP from complete oxidation of C16 fatty acid. What is C16 fatty acid? Palmitic acid. Palmitic acid. Oh, so there you are. Palmitic acid. What is? So, ATP formed from palmitic acid, 108. What is our investment? How many ATP is our investment? You have to invest. Huh? Because you have to invest. Huh? 2. There you are. Oh, ATP use. Hala. Di lagi magtanaw. Total ATP use. There, 2. So, what is our net amount of ATP? Negosyante man mo, baby. Sige. What is the net? What do you mean by net?
106.
106. 106. 108. Mauna imuhang kita minus imong investment nga 2. Your ginansya, your gain is 106. Huh? For 1 palmitic acid. O glucose pa na, asus kaguti ay. Diba? Peroxisomes oxidize very long chain fatty acids. So, mitochondrial, only about 18 carbons. Longer than that is in the peroxisomes. Breakdown of very long chain fatty acids with the formation of acetyl-CoA and hydrogen peroxide, which is broken down by a catalase, which produces water and oxygen. This system is not linked directly to the phosphorylation and generation of ATP, and also does not attack shorter chain. So shorter chain do not go to the peroxisomes. The beta oxidation sequence ends at octanoyl-CoA. So what is octanoyl? How many carbons is this?
8.
8 carbons. And then the acetyl-CoA that is produced are oxidized in the mitochondrion. Peroxisome enzymes are induced by a high-fat diet, and in some species by hypolipidemic drugs such as clofibrate. Another role of peroxisomal beta oxidation is to shorten the side chain of cholesterol in bile acid formation. No? So peroxisomes also take part in the synthesis of ether glycerolipids, glycerol, cholesterol, and dolicol. Dolicol is like rubber. So it uses fatty acids with carbon 20 and carbon 22. But this is not, of course, so much in our ordinary food. It's not. Oxidation of unsaturated fatty acid. All right. You know, there are, what we have with the beta oxidation is what we call your saturated. There's no double bond. So you can easily cut it. No. Now, if you have a double bond, especially if it is a cis double bond creates a bent structure. No, the more cis double bond, the more bent structures that you'll find. And so, in order to solve this so that it will be straight, not bent, is to transform the cis into a trans so that it may look like a saturated fatty acid. It is straight. Okay. Oh, here you are. You have two double bonds. There you are. So you cut this off. How many acetyl-CoAs? 3 acetyl-CoAs can be done through beta oxidation. And now you come up with this one. That is the beta carbon there. That's number carbon 3. And you have a double bond. So you are going to change this into an isomerase and becomes now a trans. In that way, you can cut off 2 acetyl-CoAs and then you come up with this one here. All right. So you have a dehydrogenase. Oh, as you can see that you also have a generation of utilization of NADPH here in order to hydrogenate these 2 carbons so that there won't be any double bond anymore. Okay, and so a result, you have carbon 2 and carbon 3, that's the beta carbon on top. Now you can just cut this off. And 4 ATPs, 5 acetyl-CoAs can be formed. So the intervention of having this into a trans here you cannot remove this because that's the alpha there, that's the beta. Unlike this one here, no, where you have your number 3 carbon here or the beta, so you can just cut it off 2 carbons at a time. Yeah? So it's just a conversion of that from a cis to trans. Ketogenesis occurs when there is a high rate of fatty acid oxidation in the liver. Now, conditions where you have a high fatty acid oxidation in the liver is fasting, acute, and uncontrolled diabetes mellitus. So those are the conditions that you will have a high oxidation of fatty acids in the liver. Okay, and you are going to form acetoacetate, hydroxybutyrate, and acetone as ketone bodies. All right, and this is how you form now. Acetoacetate, once you have this, it decarboxylates spontaneously, no enzyme needed, very unstable, and becomes acetone. And this is valve off through the lungs. So those patients with ketoacidosis will smell acetone in their breath. Very similar to a drunken person, no? Where you will smell acetone smell, or in the street language you call it amoy chico. No? Have you heard of that term, amoy? Amoy is what, man? Odor, no? Chico, what is chico? Well, in Spanish, chico is child, no? Male child. But we have a chico fruit. You smell your chico, especially the very ripe one, and it smells acetone. No? That's acetone breath. Acetone breath. Ketoacidosis. Okay. That acetoacetate and hydroxybutyrate is a reversible reaction, whichever is greater. When you have so much of this it become acetoacetate. When you have so much of this, it goes formation hydroxybutyrate through your enzyme dehydrogenase, hydroxybutyrate dehydrogenase. Huh? When hydroxybutyrate becomes acetoacetate, you form NADH, and NADH goes into your, where does it go?
Electron transport chain.
All right, electron transport chain or your respiratory chain. No? Okay. Now this is how you are going to excrete your ketone bodies. This is formed in the liver, and this is dumped into the blood. So in the blood, it goes into the lungs, valve off, as a gas, acetone, or goes into your urine, AMO. All right. In tissues that can activate the ketone bodies, and they can be used as a fuel. Like the brain can use ketone bodies. The heart can also use ketone bodies. And converting it into acetyl-CoA, acetyl-CoA goes into the citric acid cycle, releases energy. The producer of your ketone body is the liver. Now, from fatty acid CoA or acyl-CoA through beta oxidation, you will have plenty of acetyl-CoA. This acetyl-CoA in theory accumulates already because now we know that your acetyl-CoA is utilized in the citric acid cycle. But if this is so much already that it is saturating the citric acid cycle, there are plenty of acetyl-CoA. And this is transformed into ketone bodies and then dumped into the blood. Okay. Now, these ketone bodies here in other tissues are extrahepatic, can be utilized as a fuel through the citric acid cycle. Why can't these ketone bodies in the liver be used as a fuel? It is the producer of ketone bodies, and yet it cannot use ketone bodies as fuel. Extrahepatic tissues, that's why it's called extrahepatic. So tissues not including the liver. What is lacking here? What does these tissues have that the liver do not have? The enzyme that activates the ketone bodies. It does not have that enzyme. While other tissues have that enzyme that convert these ketone bodies, activate the ketone bodies so that it can be utilized in the Krebs cycle. So there is no activating enzyme in the liver. So acetoacetyl-CoA is the substrate for ketogenesis. That is how it is used. Now let's look at okay. Ketone body serves as fuel for extrahepatic tissues, but not of the liver, because the liver does not have this enzyme. Succinyl-CoA acetoacetate-CoA transferase, meaning to say the coenzyme A from succinyl-CoA can be transferred to acetoacetate. So CoA is transferred to succinyl-CoA to form acetoacetyl-CoA. And this can transform into 2 acetyl-CoAs. Okay, so that is the answer. The liver cannot activate the acetoacetate, but other tissues have this enzyme, succinyl-CoA acetoacetate-CoA transferase. So the CoA coming from succinyl-CoA is given to acetoacetate to become acetoacetyl-CoA. All right, so how is ketone bodies formed? All right, you have acyl-CoA or fatty acid-CoA through beta oxidation. You'll have plenty of acetyl-CoAs. Okay, and the last 4 carbons of beta oxidation is acetoacetyl-CoA. 4 carbons. Okay, now when there's so much already of acetyl-CoAs that will be formed, the last 2 carbons, the last 4 carbons of beta oxidation in the fatty acid oxidation will coalesce with a molecule of acetyl-CoA to form 3-hydroxy-3-methyl-glycerol-CoA, or what we call HMG-CoA. Now, I want you to remember this compound, HMG-CoA, hydroxymethyl-glycerol-CoA, because this compound will appear in our cholesterol synthesis. So what is it made of? Acetoacetyl-CoA plus a molecule of acetyl-CoA. Okay, catalyzed by HMG-CoA synthase, form HMG-CoA. And that HMG-CoA is split by Eliase, HMG-CoA. And you have acetoacetate, the ketone body, and acetyl-CoA. So this acetoacetate is acted upon by this dehydrogenase and becomes hydroxybutyrate as a reversible reaction. That's the one that we have shown you kanina. And this one, spontaneous reaction, forms as acetone. Now, why does this go this way with acetoacetyl-CoA? Excuse me. Because this is already saturated. There's plenty of acetyl-CoA. That is why the last 4 carbons of the fatty acid here is not converted to acetyl-CoA.